Good morning, everybody. My name is Samuel Lamon, and the title of my poster is Reinventing Peanut: Origin, Evolution, and Domestication of Arachis ipaënsis, Arachis duranensis, Neopolyploids. Peanut is an allotetraploid species with 40 chromosome number and a AABB genome type composition that originated less than 10,000 years ago from the spontaneous hybridization and whole genome duplication of two diploid wild Arachis species, Arachis duranensis, which has an AA genome composition and yellow flowers, and Arachis ipaënsis, which has a BB genome composition and orange flowers. Given the fact that peanut originated from only these two wild diploid Arachis species, and the fact that it is an allopolyploid while the rest of the genus is diploid, made peanut genetically isolated, and for this reason it has a reduced genetic diversity. However, during the polyploidization event, there has been a genetic shock due to the merging of two different genomes, which result in a genetic and epigenetic modification in the peanut genome and gene expression alteration. This created genetic instability phenomena in the peanut genome and increased the genotypic and phenotypic variability of peanut, conferring him plasticity and adaptability characteristics which are typical of polyploids. For this reason, throughout the years, peanut evolved morphologically very different and became the chosen species for domestication in the Arachis genus. Therefore, in this study, we are trying to recreate the evolution and domestication of peanut in controlled condition using colchicine-induced Arachis duranensis, Arachis ipaënsis, Neopolyploids, the IPADUR. With this, we want to test the phenotypic plasticity of this IPADUR population and investigate the genetic mechanisms that are causing this increased genomic and phenotypic variability. Here in the A image, you can see peanut flowers with different colors. Typically, peanut has orange flower, but you see that the IPADUR material is very, very diverse. What is really interesting is that these different phenotypes are not just among different IPADUR plants, but also within the same plant we can see these different phenotypes. We transition from one phenotype to the other. The B, the C, and D image are IPADUR plants where you can see have a different growing habit and different leaf coloration. The differences in leaf coloration are not due to differences in growing stages of the leaves, but they're due properly to this variability in phenotypes of this population. Some genotypes have really rare phenotypic variation, and you can notice this in the figure 1e, where we have flowers with modified shape. So this material is really, really diverse and is really, really interesting to investigate the early evolution of peanut. So we have grown this material for about 10 generations in the greenhouse, and it is showing variability not only for the traits that I've talked about, but for several others that have agronomic relevance. The one that are showing in extremely phenotypic variability, we genotype them with the Affymetric Axiom Arachis SNP version 2, and also to test the phenotypic plasticity of this material, we selected for contrasting phenotype such as light seed weight and heavy seed weight for about 3 generations. So here on the left, figure 2, you can see a color map output of the genotyping with the SNP chip. The first column is showing the chromosome number, which is the chromosome B5. The second column is showing the marker position, and then the last two columns are showing different sample genotype from plant Ipadur number 1 and Ipadur number 2. So the Ipadur plant number 1, we collected 6 samples from different branches of the same plant, while for the Ipadur number 2, we collected 4 leaf samples from different branches of the Ipadur number 2. What we will expect is when we are genotyping these different samples is to see something like in Ipadur number 1, where the genotypic code are all the same, since the plant is the branches are all from the same plant. However, some material like the Ipadur number 2 is showing different allelic code for samples from the same plant. This is called allelic mosaicism and is implying something is occurring at a mitotic level with this material. We will further investigate what is occurring genetically using Illumina sequencing. The images in the center, you see the selection for contrasting phenotype for light seed weight and heavy seed weight. These are the pinose pods of Ipadur plants just after 3 generations of selection. You can definitely see that there is a difference in size just after only 3 generations of selection, indicating the increased phenotypic plasticity of this material. On your right, you can see violin plots for plants selected for this contrasting phenotype. So on the left, the trait investigated is seed weight. On the right, the trait investigated is the same number. In blue, you can see the violin plot of Ipadur plant that were unselected. In pink, Ipadur plant that were selected for heavy seed weight. And in green, Ipadur plant that were selected for light seed weight. The first row is representing Ipadur plant data after the first generation of selection. The second row, after the second generation of selection. The third row after the third generation of selection. And the last row is combined data from the beginning of the selection to the current point. So you can see that the pattern in terms of seed weight doesn't change in the 3 generation. We have statistical difference between the unselected material and the light seed weight, while something is changing in terms of seed number from in the 3rd generation. Usually we will have no statistical difference between the 3 different selection. However, in the after the 3rd generation of selection, we see that some of the seed selected for heavy seed weight, they have an increased seed number and they are statistically different from the unselected material. This is really cool because this means that we have seeds selected for heavy seed weight, which also produce a lot of seed. So this is potential, there are potential genotype for germplasm release. If we combine the data from the beginning of the selection to the end, we will notice that there is statistical difference between all the 3 different treatments. So between the unselected Ipadur plants, the Ipadur plants selected for heavy seed weight and for light seed weight. And as regards the seed number, it has a similar situation for what is shown after the 3rd generation of selection. Therefore, this Ipadur population has a lot of phenotypic and genotypic diversity, and it has a lot of potential to introduce new alleles for agronomical relevant trait in a peanut crop. And with this experiment, we are investigating the early evolution and domestication process of peanut. The plants that are showing increased genotypic diversity, we will further investigate them using Illumina sequencing, and just after 3 generation of selection, we are able to have statistical difference among the ipadur plants selected for in different direction. Eventually, we will proceed with this material, performing era trials in order to calculate the realized genetic gain over selection of this material. With this, I want to thank you for your attention.